This study uses Piankov's publication of the tomb of Ramses VI, an English translation that Eric Horner claims has yet to be superseded, supplemented by Budge's illustration of the tomb of Seti I for the following reason. According to Piankov, complete versions of the book Amduat, which first appeared in the 18th dynasty, are found in the tombs of Tutmosis III and of Amenapis II. The first 11 divisions are found in the 19th dynasty tomb of Seti I and in the 20th dynasty tomb of Ramesses VI. Piankov's notes that the best version is from the tomb of Seti, and that the version from Ramesses VI is abridged, and registers of divisions 7, 8, 9, 10, and 11 are hopelessly intermixed. Because of this, Piankov constructed his diagrams of the hours from 18th dynasty version of Tutmosis III to Amenapis, referencing 18th version provided continuity and a larger evidentiary base of text and images for this thesis. Also displayed are Budge's illustrations, which are traced from the plates of the tomb of Seti the first and presented in his analysis the Egyptian heaven and hell, a 1905 text now in the public domain. Budge's translation will be used when appropriate. The Hidden Chamber. Of all the funerary texts, the Amduat of the Book of the Hidden Chamber explains the central Egyptian doctrine of life after death related to the transfiguration of the deceased. The book describes quantum events related to the lytic cycle and the excitation of the Lambda prophet, which is Osiris, by mapping the preferred path from this world to the next that was only available for souls who rode in Ra's ferry boat, that is, the Lambda vector. In the afterlife, the text exhorts the deceased to go forth into the day, or to join the light of the sun. After merging into the sun god, the deceased is transfigured into the shape of the being god, a being of matter and light in the energy funnel of the Duat. But to make this transformation, the deceased must travel in Razbak through the dark, deep valley of the Duat, along a guided diffusion pathway of incoming phase lambda through the Lamporn to the site of the integrated lambda genome on the whole cell chromosome at the LAC-Z or B-gal gene seats. The chemical journey through the Duat symbolizes or resembles the thermodynamic energy landscape of the host cell, where proteins fold to native formations within an energy landscape funnel. In this process, the deceased visits the hidden chambers of Osiris. The 30 BP prophet attachment site contains a conserved central 15 BP region where recombination takes place. Similarly, the domain of Osiris contains 15 Ots and the Egyptian afterlife game. Senate had 30 squares, suggesting the 30 BP prophet site of the phage lambda. The significance of lambda as 30 is expressed by Greek lambda / equaling 30, which probably stemmed from the Hebrew lamed equaling 30. Also, text reference the house of 30, allusions to 30 BP lambda, attachment site on the host chromosome. The sun god's journey results in the excision of the prophet and rolling circle replication of crow protein production of human phage progeny, the Haas species. DNA to RNA to protein. Like the supramolecular chemists identifying molecules Numerous deities in the Andua are able to distinguish each other by their shape or properties. Thus, we have the pharaohs using a new science method to describe quantum experiences and molecules that the deceased encounters in the afterlife. The elaborate Egyptian system of metasymbols depicts the shape and properties of molecules in the universal catalytic process, binding of the substrate, chemical transformation in the product, and 3, release of the product to the regenerative, the active catalyst, or to regenerate the active catalyst. Sorry. The 12-hour journey of the deceased god, sun god, and crew into the Dua or earth cell represents the catalytic process and follows the central dogma of DNA to RNA to protein. The presence of the deceased at the northern pole of the earth cell is important, for polar localization of the proteins is critical for bacteria cell division, chromosome partitioning, and the cell cycle control. In the first stage of absorption, phase lambda binds at the maltose receptor site, LAM, of the bacteria earth cell. Induction of the maltose binding protein activates the formation of a polar distortion called a polar cap. No, you can take that. That's what I'm talking about. See? That's what I'm talking about. All right, shit.
Can't go nowhere.
Can't go no goddamn way.
Right?
Can't wait to get my leather jacket with my fox fur. So that's where I'm coming. Right in this
Gotta wait
motherfucker.
to work.
Yep. I'm gonna do like this when I hit the door too.
That's right. Hi y'all. Come here. Check me out.
Lex A monster repressor protein Apophis. The pyramid text refers to Apophis as a full-coiled monster snake. The structure of Lex A repressor, as determined by IH and NMR spectroscopy, contains 3 regular alpha helices and 1 anti-parallel beta sheet. The beta sheet exhibits a coil-like shape that could be described as a coil. Thus, we have a full-coiled monster that represses 42 genes while allowing the active lambda CI repressor to maintain the prophets in an inert state and permitting the active LAC repressor protein to bind to the DNA operator sequence to prevent lactose activity. In hour 7 of the Amduat, and we find textual references to Apophis and a drawing of the long-feathered serpent. Rec A protein Mahin. Rec A is a multifunctional enzyme with 2 roles. In its first role, the protein mediates strand exchange during recombination. The second role involves the cleavage of the Lex A repressor protein Apophis during an SOS response. Rec A protein has a smaller domain, stabilizing a 6 helical polymer of protein subunits in the interhelical bundles. Representation in the Cairo coffin CGC 28083 map from the Book of Two Ways depicts a smaller structure, which is depicted in figure 15.3a. The top op UA represents the 2 smaller domains stabilizing the 6 helical polymer of protein subunits signified by the bottom 6 UA of interhelical bundles. However, in the Amduat, the protein is depicted as the serpent around the sun god, which seems appropriate for in its first role related to recombination. Rec A forms a helical filament around ssDNA. In a co-protease activity, the ssDNA/Rec A filament cleaves the Lex A repressor protein, which catalyzes its own digestion. Number 3, crow repressor protein horse. Hour 4 of the Amduat shows a 3-headed serpent that matches up with the active 3 helix crow repressor of phage lambda, with its 3 helices. The DNA binding protein in 15.4a is a homodimer, with each subunit being 66 amino acids in length. Take a look at these pictures here. I know I showed them earlier, but we glimpse at it now. Alright, they really breaking this down. Having a helix turn helix motif, the repressor has a 3 alpha helices, 2 of which are separated by a short turn with a 3rd helix filling into the major groove operator DNA. Figure 15.4a depicts a sketch of the active crow protein with 3 serpent heads, 14 discs, 14 heads, 14 stars from Amduat hour 4 in the tomb of Ramesses VI. Lambda CI repressor protein Seth, which is seen in figure 15.4b, regulates phage gene expression in the lysogenic stage by binding to OR or OL operator sequences. The dimeric repressor has 5 alpha helices in a bundle with 2 helices and 3 forming a helix turn helix. One can easily equate the image of the 5-headed serpent repressing the inert Osiris in the Amduat hour 6 with the 5 helix lambda CI repressor protein. Catabolite gene activator protein CAP. The winged 3-headed serpent in the hour 5 represents CAP protein, also known as CAMP repressor protein, or CRP, classified by modern biologists as a winged helix turn helix DNA binding protein. The protein exhibits an extensive beta sheet structure which are wings, and the 3 alpha helices, which are heads. In the absence of glucose, Cat binds to the CRP site near the lac operon promoter, stimulates transcription of the lac operon, and interacts with RNA polymerase.